That's done. Now I had, now, okay, yeah. So, yeah, sorry about that. Yeah. Hi everyone, I'm Pol Sola. I come from Barcelona. I will be presenting this method on the poster J11. This method is aimed to be a quality control algorithm for LC-MS signals, okay, because what we have normally when we have this type of experiment, what we usually have is that when we receive this data set, we make a PCA and we start to plug in here all the variances that we know. For example, we have batch effects, drifts, we have the blanks, and for example, in red there is, we were lucky and we have, for example, the concentration of the samples that we usually not get for example, for a urinary data set. But what happens if we do not have anything about this information, if we do not know those uncontrolled sources of variance in the data set? This is what we are trying to tackle in here. So what we do is actually to order the chromatograms in the injection order of the equipment, and then through a sliding window, trying to characterize sample by sample in a quantitative and qualitative way to actually depict what are the problems and the events present in the data set. Let me put you an example about that. For example, if we want to analyze a sample, what we will do is to take the previous 10 or 15 samples and build a principal component analysis. Take the first principal components, which are explaining the most of the variance of the previous samples, and project a new sample. With that, what we know is that basically if you fall out of the plane, the new sample, what we'll have is a new variance. So this error is called the Q statistic. For example, in a change of batch, this will happen. The other error that you can commit is that when you project the sample on the plane, that it falls from the normal deviation. This could be, for example, due to a phenotype or due to the concentration of the sample. By combining both, we can actually characterize and qualitative and quantitatively the events on the whole experiment. For the results of that we have validated through 2 experiments, please visit my poster on J11. Thank you.